TCR junctional diversity, or in-region addition. Please remember, irrespective of organization, the 12-23 rule for RSSs must be followed. An Ig heavy chain DNA recombination recognition signal sequence may look like this, or sound like this. L, VH, 23, 12, DH, RSS1, 12, RSS2, 23, JH, CH. Remember VDJ joining in immunoglobulin. Location and organization of DNA RSS of delta chain germline TCR DNA is the same organization for beta chain. You have L, V delta H, 23, N, 12, H, D delta H, 23, N, 12, H, D delta H, 23, N, 12, H, J delta, C delta, 3 prime. Remember that VD, DJ, and the C stands for the constant portion. For the N-region addition, V delta, J delta, 5,461 to the first power, alpha, beta, gamma, and delta chains. V delta, D delta, J delta, 5,461 squared, beta, and delta chains. V delta, D delta, D delta, J delta, 5,461 to the third delta, and beta chains, but only seldom. So we're talking about the germ lines that this can occur in. The alpha, beta, gamma, and delta chains can all have V delta, J delta, or V to J joining, but their addition would be 5,461 to the first power. The VDJ, or V delta, D delta, J delta that can occurs in the beta and delta chains. They have 5,461 to the second possibilities of joining. The V delta, D delta, D delta, J delta, or VDDJ joinings occur in the delta mostly, but can sometimes in the beta, but it's very seldom. They have a combination possibility of 5,461 to the third. Note, V beta and V delta can associate only with J beta or J delta respectively. Alpha and gamma do not have D segments. Alpha chains and gamma chains do not have J segments. Let's try to estimate the number of human TCR. If you have an element, say, in your V variable segment of alpha, you would have 50. If you have a variable portion of your beta chain, you would have 57. In your alpha chain, how much would you have in your D segment or your diversity segment? You should have said 0. We just said that there are no D segments in alpha chains. The diversity that's possible in a beta chain D segment is 2. D segments in all 3 frames. How about in the alpha chain? Once again, alpha chains do not have D segments. That is reserved for beta and delta chains only. In the beta chain, the D segment occurs in all 3 frames very often. Let's talk about alternative joining of D segments. In the alpha chain, you would expect how many? Once again, alpha chains do not have D segments. That is reserved for beta and delta chains only. For a beta chain, the element of alternative joining of the D segment does occur. The J segments, or the joining section of an alpha chain in TCR, has 70. In the beta chain, the joining segment has 13. Junctional flexibility occurs in both the alpha and the beta TCR. Joints with N and P nucleotide addition, alpha chain has one joint that has a possibility of N and P nucleotide addition. The beta chain of a TCR has 2. Sometimes 4 because of the possibility of D to D joining. In the alpha chain, the P and nucleotide additions occur in V and J. In the beta chain, you have the possibility of it happening in V and J, VD and J, VD, D and J, and even possibly VD, D and J, but it is very, very seldom. Combinatorial association of chains. Alpha chains and beta chains both have combinatorial association of chains. Number of V or variable gene pairs, around 5.8 to the 6th for both alpha and beta TCR. Junctional diversity is about 10 to the 13 for both alpha and beta TCR. Total diversity for alpha and beta TCR is approximately 10 to the 19. Let's take a moment to talk about the TCR CD3Z complex. In association with the TCR, part of the TCR intracellular signaling complex within the cytoplasmic membrane is then complex called CD3. The CD3 complex lies within 12 angstroms of the TCR. The CD3 complex is somewhat analogous to the Ig alpha beta protein complex in association with the B cell's antigen receptor Ig. The CD3 protein complex consists of 5 different polypeptide chains: gamma, delta, epsilon, nu, and zeta, which associate in various combinations to form 3 dimeric molecules. These 3 dimeric molecules exist at 1 as a homodimer of 2 identical disulfide bonded Z polypeptides, or sometimes a heterodimer of the Z-nu combination. 2, a heterodimer comprised of 2 non-covalently associated polypeptides, 1 gamma and 1 epsilon. And 3, another non-covalently associated heterodimer that consists of epsilon and 1 delta polypeptide. Let's talk about the accessory molecules and T-cell activation. Many interactions in the immune system require physical cell-to-cell contact in addition to cytokines. Developing B and T cell precursors require intimate contact with stromal cells. In the generation of immune responses, there are at least 3 critical cell-to-cell contact events required. The first is when a naive T cell initially contacts its specific antigen. Because of the requirement for MHC presentation, this is an obligate cell contact event. In normal situations, the critical antigen-presenting cell, or APC, is the dendritic cell. In addition to the MHC peptide TCR interaction, there are other non-antigen-specific membrane-bound ligand receptor pairs that are important for the APC T-cell interaction. The principal one is the association of the CD28 molecule on the T cell with either of 2 ligands, B7-1 or CD80 and B7-2, CD86. Either of these molecules on the APC have a principal association with the CD28 molecule on the T cell. These molecules are called accessory molecules, and we know that the CD28 molecule delivers an essential second signal to the T cell without which the T cell does not become activated. In fact, it becomes unresponsive. The second essential cell-to-cell contact is between the activated T cell and an antigen-specific B cell. Most antigens are T-dependent. The antibody response to them absolutely requires T cell help. This help is delivered by both cytokines and cell-to-cell contact. B cells bind specific antigen by their surface immunoglobulin, endocytose, process, and present it in their class II MHC molecules. This enables them to be recognized by T cells specific for helper epitopes from their specific antigen. This cell-to-cell interaction also requires CD28 binding to B7 on the B cell. In addition, activation of the T cell has induced expression of a molecule called CD40 ligand, or CD40 that binds to CD40 on B cells. Cross-linking CD40 promotes B cell proliferation, prevents apoptosis of germinal center B cells, and promotes antibody class switching. Blocking the CD40-CD40L ligand interaction leads to B cells that cannot proliferate or produce antibody in response to T cells. I'll repeat that again. Blocking the CD40 interaction with CD40 ligand leads to B cells that cannot proliferate or produce antibody in response to T cells. The CD28-B7 and CD40-CD40 ligand receptor interactions are both essential for the dialogue between B and T cells that leads to their mutual activation. They need each other. The third cell-to-cell interaction essential in immune responses is the binding of activated B cells, which have migrated into a specialized structure in lymphoid organs called germinal centers, to follicular dendritic cells, or FDCs. FDCs are specialized stromal cells that hold intact, or, in other words, unprocessed antigen on their surface in the form of long-lived immune complexes. Among other molecules, FDC express CD23 that binds to germinal center B cells by their CR2 receptor and stimulate differentiation into plasma cells. There are a lot of accessory molecules, and we're going to go through a couple here. CD4 and CD8. They are T cell co-receptors for the recognition of peptide MHC on antigen-presenting cells, which bind to the non-polymorphic regions of class II and class I MHC molecules respectively. They both promote the adhesion of antigen-presenting cells and targets and early signal transduction events. Along with CD4 and CD8, you have CD2 or LFA2 and CD58 or LFA3. CD2 is an accessory molecule that acts as an adhesion molecule and signal transducer. CD58 is an accessory molecule that acts as an adhesion molecule and signal transducer as well. It is the ligand for CD2. CD11A, CD18, also known as LFA1, CD54, known as ICAM1, and CD102, known as ICAM2. LFA1 is an integrin adhesion molecule. Its ligands are ICAM1 and 2. CD28 with ligands B71 and B72. CTLA4 ligands are also B71 and B72. CD28 is a co-stimulatory molecule expressed by T cells that transduces signals together with signals delivered by TCR complex-mediated activation. CTLA4, or CD152, is expressed on recently activated T cells that functions to inhibit T cell activation by counteracting CD28 cells, or CD28 signals. I'll say that again. CTLA4, or CD152, is expressed on recently activated T cells that function to inhibit T cell activation by counteracting CD28 signals. We're going to talk about the CD40 ligand for just a second. CD40 ligand, or also known as CD154, activates B cells, macrophages, and endothelial cells. It is the ligand for CD40. CD45. A cytoplasmic tyrosine phosphatase plays a role in T cell activation. Different isoforms of CD45 are associated with antigen-naive versus memory T cells. Those are the accessory molecules that you need to know for now, and for now, it's enough. Let's move on. T cell activation. Non-transmembrane protein tyrosine kinases are involved in that. The role phospholipase C. You really need to figure out what the role of phospholipase C is. I'll let you take a moment for you to remember.